And so that's what's most commonly used would be sterile water for flushing. And then you don't run into those issues of, is my detergent, does it have a lot of particulate matter in it? Is it very hard water that could potentially clog nozzles on the scope? So that's why we recommend that the sterile water be used.
What if one conversation could save a life? What if one new process or new product could save a patient from surgical harm? Broadcasting from a hospital basement near you, you are listening to the Beyond Clean podcast, the global voice of sterile processing. Each week we spotlight the best practices, products and people from around the world who are disrupting our profession and helping you fight dirty. Every instrument, every time. Join us as we have the kinds of conversation that matter and give our industry the kind of voice that it deserves. Nyt on aika mennä puhdastakin puhtaammaksi. And now it's time to go beyond clean.
This week on Beyond Clean, we speak with Marianne Drosnock, Director of Clinical Affairs at Healthmark Industries, and we're going to be talking about all the chemicals that are used in scope reprocessing, specifically endoscopes and GI endoscopes, Hank. This is going to be a great conversation. Marianne has proven to be a phenomenal guest on the Beyond Clean podcast in the past, and so not to get everybody's expectations up really high, but why don't you go ahead and do it? This is going to be a great episode.
Yeah, Justin, you hear the term expert kind of thrown around a lot in our industry and healthcare in general, but Marianne is the expert and is a fantastic expert to be talking about the topics that we're going to be talking about today, chemicals, and not only that, the updates, the guidelines. So it's not just like, oh yeah, she knows the guidelines really well. Like she is thoroughly involved, co-chairing the committees and the work groups that are putting these babies together. So there is no better person to really dive into the context for how these things are being written and why they're being written the way that they are. So I'm pumped. It's going to be a fantastic show, and I have a feeling it's going to be another blockbuster.
All right. Don't go anywhere. We're going to be right back with Marianne Drosnock, Director of Clinical Affairs at Healthmark Industries. A long-awaited announcement has come from OneTray stating that the FDA acknowledges that OneTray may be stored for up to 48 hours following sterile processing. The FDA also confirmed that IUSS containers cannot be stored. The OneTray, therefore, is not an IUSS container. There has been a lot of confusion surrounding the warning letter, and OneTray offers full support to anyone that has any questions. Patient safety is number one, and it is encouraged to always reach out to the manufacturer if you have any questions regarding a product. If you would like more information on their journey with the FDA, subscribe to www.onetray.com/subscribe. If you would like to reach out to OneTray directly, you can email info@onetray.com or call 937-619-0138. Follow the OneTray IFU for use. Help support patient safety with 3M chemical indicators and be more confident of sterilization results with 3M innovations. 3M Attest vaporized hydrogen peroxide trimetric chemical indicators are the only FDA-cleared VH202 CIs to independently monitor three variables: exposure time, temperature, and concentration. 3M Attest steam chemical integrators are designed to be easy to find and quick to read, streamlining workflow so your team can focus on patient safety. See more at go.3m.com/3MCI. That's all lowercase letters, go.3m.com/3MCI. I'm Justin Poulin.
And I'm Hank Balch.
From the 17 studios, you're listening to Beyond Clean, the global voice of sterile processing.
Joining us now is Marianne Drosnock, Director of Clinical Affairs at Healthmark Industries, and also the most downloaded podcast guest in Beyond Clean history to date. So we're pumping you up, Marianne, before we even say hello. But, we're going to put this episode up against your prior one and see which one wins out over time.
No pressure there. Yeah. Well, thanks for having me.
So, you've been up to quite a bit, and I know we've been kind of had this conversation on deck for a while now as we were waiting for some updates to ST91. And we're really going to talk about scoping in our chemicals, as Hank has this one titled. So before we get started, I would think that the most downloaded guest in Beyond Clean history wouldn't need to introduce themselves, but just in case nobody is familiar with who you are and what you've done in the industry, can you talk about your experience to kick us off?
Sure. Just a brief background. I don't want to take up too much time with it. My name is Marianne Drosnock, as the introduction said, and I'm so happy to be here tonight. I'm the Director of Clinical Affairs at Healthmark Industries, where I lead a team of educators that help provide best practices and knowledge related to reprocessing and infection prevention to all of you out there in this listening world. And I'm so happy to be here tonight to impart more knowledge and updates regarding chemicals and a little bit about the new ST91. And I am just so excited.
Excellent. Well, we're excited to have you back. And when we talk about, scope reprocessing and chemicals, right? Like, where do you think we should start with that conversation? What makes it maybe different for scopes than regular stainless steel instruments?
Well, I think that's a great question. And although it may seem obvious to a lot of people, but I think it's worth the explanation that flexible endoscopes and other types of scopes, rigid scopes in particular, or other stainless steel instruments, they're different, right? They can't be subjected to the same type of chemicals. They can't necessarily go through the same kinds of sterilization processes because they're fragile. They're much more susceptible to damage. They have coatings on them and adhesives that can wear away with exposure to those different oxidative chemistries and heat. And that's why we don't have a lot of products in the endoscope world, at least the flexible side of things, that are compatible with steam sterilization. In an ideal world, that would be great, right? I would love to be able to say we can steam sterilize these devices, but realistically, there's very few on the market, only really a handful that could be steam sterilized, and those never really caught on very much. So that's the major difference I see between our stainless steel instruments is you can't put them through the same processes. We still have to resort to high level disinfection, which is a whole nother realm of chemicals we're going to talk about. And although, we want to move to sterilization for these devices, they have their special circumstances that we have to really keep in the forefront of our mind.
Can we talk about the categories of chemicals now, Marianne? Like you just alluded to high level disinfection. So that's one of the categories that we're going to talk about. But can you kind of lay out, from the very beginning, or I don't know if this is the beginning because it's a cycle, but at the point of use all the way through this process, what kind of chemicals are we encountering in that workflow?
Yeah. So that's a great question. And I think we'll have to start all the way back at the beginning of the process with detergents, because we know immediately after the procedure is done, we start that pre-cleaning process, or what we used to call pre-cleaning that we're going to now call a treatment, pre-treatment, so that we can further prepare that scope for processing in sterile processing or the SPD department or endoscope reprocessing area. But we do that step of pre-cleaning with a detergent solution normally. And what we're looking for is just something general, and this is what ST91 has said and will continue to say, is neutral pH, low foaming, and formulated for medical devices, specifically endoscopes, in what we're talking about tonight. So that's what you're looking for as far as the detergent in that first step of the process, which is always meant to be done in the procedure room. It's a very basic process, wiping down the outside of the scope with that detergent solution that's been soaked onto a sponge or a non-linting cloth that you wipe it down with, and then flushing the channels or suctioning them with the detergent solution. Now, some endoscope manufacturers have validated water for that pre-cleaning process, and that's okay if your facility decides to use water per the IFU and per your internal policies for those types of scopes. But most facilities still use detergent for that step, and that's really what we're looking for, is that basic category, low foaming, neutral pH, formulated for medical devices. So that's that first basic category of chemicals that you'd be looking at in endoscope processing.
And now you mentioned water, and I think we've covered this maybe once or twice on a previous episode, but I want to ask kind of a dumb question here. Technically, water is a chemical, right?
Correct.
Okay.
It is.
Okay,
that's right. Yeah, it can be considered a basic cleaning chemistry. It's really well suited to flush the channels. You don't worry about compatibility issues. You don't worry about incompatibilities between that detergent and a detergent that would be used at a later step in the process during manual cleaning. You don't worry about having to rinse that residue off before you go on to the next step. So there are benefits to using water, but a lot of facilities don't necessarily feel comfortable with that, even though the IFU says you can do it. So it's a matter of making that internal assessment, risk assessment in your facility to know what's right. But you are getting rid of a detergent and a cost then by not using it during the pre-cleaning process.
So when water is used, I hate to get off on this tangent for water, but I'm kind of curious about this. So is it always sterile water, or do they just kind of leave that open for interpretation in the IFU?
Well, because you're at the step where it's just cleaning, it's that basic pre-treatment. It wouldn't necessarily have to be sterile water if you think about it, but that would be recommended, right? Because you're going to be using sterile water during the procedure, so you have it right there. And so that's what's most commonly used, would be sterile water for flushing. And then you don't run into those issues of, is my detergent, does it have a lot of particulate matter in it?
Right.
Is it very hard water that could potentially clog nozzles on the scope? So that's why we recommend that the sterile water be used.
I've got the voice of Jonathan Wilder like over my shoulder whispering in my ear about water. So I'm just like, I better follow up on that question. That's an interesting point. Detergents, all right? So you set the stage, that's category number one. That kind of gets us kicked off. What's coming after that step?
Yeah. So after we do that pre-treatment step in the procedure room, we transport it to wherever that scope is going to be further processed, normally sterile processing or endoscopy, reprocessing area, or it might be to an off-site location. Who knows? Everybody's hospital is configured differently. As we say, if you've seen one hospital, you've seen one hospital. So wherever that scope is going to be further processed, we do that, containerizing that scope. It comes into the reprocessing area. It'll be leak tested. And then the next stage of processing is manual cleaning. And manual cleaning is always performed in that detergent solution. So it has to meet the same characteristics as we said for those used during the pre-treatment phase of it. So neutral pH, low foaming, formulated for use with endoscopes. We can't use water only in this stage. So that's not acceptable. So you have to think about it, logistically. You may use water or detergent during pre-cleaning. You're always going to use water during leak testing, and you're always going to use detergent during manual cleaning. And sometimes I see detergent used during leak test. It should never be. It should always be clear water for that. And then you want to add your detergent for the manual cleaning stage of it. So same characteristics of the detergent, some helpful hints. You really want to drill down and look at that dilution of the detergent solution and make sure that it's being diluted appropriately. So according to the label of the detergent bottle that you have, will tell you it might be 1 ounce per gallon or 1 ounce per 3 gallons, whatever it is. And that should be measured out because you want to ensure you have that proper dilution. If not, it's not going to be strong enough. If it's an enzymatic, might not be enough enzymes there, or if it's too strong, if it's not diluted enough, then you may have problems rinsing it off, which can lead to damage on the endoscope or interference with the disinfectant in the later step or the sterilant later on. So we really want to make sure that they're properly diluted according to the instructions. The other thing you want to keep in mind is the temperature. So as ST91 states, if there is a temperature on that label for any chemical being used, then it needs to be monitored during the process. So you need to monitor that temperature, however it is that your facility wants to do it. Most facilities will have a, temperature gauge with the line that runs down into the water, or it might be a sticker that goes into the water that has the different temperatures on it. We don't state within the standards exactly how to do something, just what you should do or what you have to do. How you decide to do that is really up to your facility. But the point of all that is you want to monitor the temperature in that bath to make sure it stays within the range that's listed on the label of the detergent. So going back to that label on the detergent bottle, it might have a very broad range. It might have a very narrow range. And sometimes that guides facilities into which detergent they try, because some might have a little more flexibility. Not that that's a good or bad thing, but some have a little more flexibility. Others have a narrower range that they work because, enzymes only work at specific temperatures. So if they're too hot, they're going to get killed off. Those proteins are going to be denatured and won't work. Or if it's too cool, then they're slow down on their action and you won't get that cleaning within the right time. So dilution and temperature is so important to remember when you're doing that manual cleaning process in the sink.
So I like to ask kind of personal preference questions when we're talking to experts like you. So forgive me, I'm going to kind of put you on the spot here a little bit. You can give me a political answer if you want to. In terms of the chemical development, do you have a preference around color or smell of these chemicals? I know that's kind of a constant conversation in the industry. If you're constantly over this sink, do you want it to have a smell? Do you not want it to have a smell? Do you want to have a color? Like you mentioned, the very important point about leak testing in water only, right? If you've got a clear detergent, that could be a little challenging if you're relieving someone or just not a very good memory like me. So yeah, do you kind of land any place on that? What's your feedback around those two things?
So I can't say I have a real personal preference, but I can see the benefits and risks of both sides. As you said, if you use clear, you might not be able to tell if you actually added it. And fragrances might lead to irritations of people that are breathing these vapors in all the time. But to be honest with you, I've not heard of, specific incompatibilities or sensitivities related to any one type or another. And like you said, with the blue color or a green color might be better to see if it's been added or if something's been missed. You never know. You might see that color dripping out of the scope or something. So it could be an indicator, but personally, I don't have a preference around color or scent. What I want is what works best for cleaning the device. And there's a lot of detergents out there and a lot of them that don't have the data to back up what their claims are. So for me, the most important aspect of it is asking the manufacturer, what data can you give me to support that this is actually going to clean scopes or clean it within the time and at the temperature that you state? Or it might be that, they have a specific biofilm claim or a specific removal of simethicone claim, whatever it is. But I want the data to support that because there are test methods through ASTM that are out there that people should be testing to. And as a consumer of that product, in the hospital facility, you should be able to get some of that data to help prove that that's the detergent you should be using.
So what you're saying is you can't just believe any marketing that you see out there in the industry.
You're absolutely right.
Just wanted to confirm.
That's right.
So that question of efficacy has come up a number of times actually in this chemical season. So I'm so glad that you brought that up again, because it is, even if it is developed appropriately and it's marketed appropriately, we can still use it inappropriately in a way that decreases that efficacy. This is, you mentioned, the temperature, the dilution, all that kind of stuff has to be built into the process and kind of hardwired so that we know that it's operating the way that it was formulated to operate. So I appreciate you bringing that up. Go ahead, Justin.
And another really interesting nuance too is just the way that you process, like gastroscopes and colonoscopes, is that you fill the sink and you only use it for one reprocessing. And that's one of those areas when you talk about efficacy, Hank, just to piggyback off of that, like many people don't drain the sink when they're doing stainless steel, reprocessing of those instruments, and will do several trays going through. And at some point it does lose its efficacy just because, the chemicals that are in like a detergent all get used up. And so when you're talking about scopes, it is baked into a standard reprocessing technique that kind of eliminates that particular variation or judgment call. It also helps us, I think, maintain temperature for reprocessing so that, enzymatics especially are at that right, temperature so that they're activating and et cetera. But when we talk about standardization, and that's what I love about that scope processing kind of pathway, right? It's very standardized, at least in that one aspect that I just said. But then there's still a lot of other variations in terms of chemicals and reprocessing. Do you see any trends happening right now or any kind of movement towards even more standard reprocessing protocols?
Yeah, Justin, that's a great question. And I don't even know where to start because there's so much I could say on this. So yes, for sure. Let's start with the obvious, right? The elephant in the room. Sterilization. There is definitely a trend, and one that I am a proponent of, and I hope goes really to the next level, is sterilization of endoscopes. So I think in our heart of hearts, right, we all want scopes to be sterile. I don't think that patients know that their scope used on them isn't sterilized. And if we really think about where the Spalding classification or how it was written with endoscopes being a semi-critical device, and go back to what it said originally, is that those devices should be cleaned and sterilized. And then if that is not feasible, we resort to high-level disinfection. But over the years, as an industry, we've come to accept high-level disinfection as the standard of care for flexible endoscopes when we should have always been striving for sterilization. So this next ST91 is really going to focus on that, moving towards sterilization. Listen, we're realistic. As much as I'd love to say we can do that today, we know that's not feasible. But we want to be propelling the industry toward that, making sure that manufacturers know we want scopes that are materially compatible with sterilization, and we want sterilization modalities that are FDA cleared to sterilize endoscopes, because until that's readily available for everyone, we're not going to get there. But we really wanted to refocus in ST91 on knowing that these scopes should have always been sterilized, and they should be sterilized if that's possible. So what I see in a lot of facilities is a movement to getting those scopes that are already sterilizable to sterilization. And I know that sounds simple, but I do a lot of auditing. You go around to hospitals and I look at practices for endoscopy. One of the first things I look at is, what are you doing with your bronchoscopes? And they'll say, we're running them through high-level disinfection. And I say, well, does sterile processing have a STERRAD or a vPro system? With the right cycles within that? And they say, of course, yeah. And I said, then why aren't those scopes being sterilized when they are a semi-critical device that should be cleaned and sterilized? And those scopes are going into normally a much more ill patient population. If you don't have a screening bronchoscopy, right? You're doing that for cause. So our patients are sicker that are getting these used on them. And you have sterilization methods already there at the hospital that are compatible with reprocessing for that scope. So why wouldn't you be sterilizing it? You get a higher margin of safety built into that process. You get a wrapped item that you can store in accordance with your hospital policy for wrapped items. So why wouldn't you do that? And I know it's about giving up, maybe that department doesn't want to give up their scopes to sterile processing. They think, it's going to lengthen that turnaround time, which it may. You may have increased costs because of chemical compatibility issues, or, sterilants are generally harsher chemicals and harsher processes. So you may see repairs happening more frequently or more quickly. So, thinking, I don't want my repair budget to go up. I don't want to lose track of those scopes, but really that's the way we need to be going. So look at that low hanging fruit. And if you have scopes that are compatible with sterilization, they should be sterilized. It boils down to that. So that's the most major trend, I think, that we're going to see and are already starting to see is that move to sterilization.
What about the movement towards automation? Is there a downside towards leaving manual cleaning in the past and doing more automated reprocessing?
Well, Justin, I think, of course there is, right? If you automate a process, you take out that risk for steps being missed or being done improperly. We're human. We all have bad days. We might not have slept or our kid was up all night, whatever it may be, can make it challenging the next day. So automation can definitely help with that. So it's something that we have to be open to is different types of advances in technology as they come out and making sure that they have the right clearances and being open to that. So I definitely think automation is a part of it in the future. And the other movement that we see a lot of or trials of, I should say, is movement towards single-use endoscopes. That's a big discussion. Again, we could have a whole podcast just on that, right? There are many types of single-use disposable endoscopes that are now available. They come to you sterile. They're used once and they're disposed of or they're recycled. So I do think that that's going to be something that gains momentum and picks up speed over the next couple years. It already is in certain applications.
Yeah, Marianne, what's so complicating or complex is there's a lot of people who have a lot of vested interest in where the industry lands on these questions. If you are a manufacturer of reusable scopes, you have an opinion on the matter, and you likely have a lot of science and research to back up your opinion. Likewise, on the other side of the fence, for single use, the same is true for sterilization and high level disinfection. So it's a microcosm of the world out there in sterile processing of everyone's kind of got their own facts. At times it feels like, right? Like at the end of the day, we should be able to agree on the science, but it can be very disorienting for a department leader kind of in the middle of these competing voices coming at you saying, oh no, like, did you see my white paper? Well, or what about this new research that came out that says something completely different about this? And it's really hard to navigate. If you were to kind of speak to that person who maybe feels that way or maybe will feel that way after this episode, once they hear some more of the details, how do you comfort them? Like, where do you direct them to go to try to find something solid to decide on or, work together, I guess, within their facility to come to a conclusion?
Yeah, Hank, that's a great question. And I think one that we're all going to struggle with over the next couple of years, because what I believe is going to happen is we're going to come to some sort of equilibrium where we see a little bit of this and a little bit of that and a little bit of that. So, and what I mean is you're going to see a certain part of the market captured by single-use scopes. It might be just those that are used in the ER, or it might be those for difficult airways, or just duodenoscope, something like that. So I do believe a certain portion of that market, you'll see single-use scopes really coming to the forefront. I believe there'll always be a place for reusable scopes also in the market. So what we have to do is figure out what are we going to do with those scopes. And within that segment of the market then, like I said, we want to see things move as much as to sterilization as we can. Some of that might go for liquid chemical sterilization, but we still have those tough scopes where we don't have a lot of methods available now. So we're going to see high level disinfection hang on at least for a while for certain types of scopes. Again, maybe that's the duodenoscopes because they are a GI scope that's complex, but we know they have issues related to them. So what are we going to do to make that process better? So Hank, I don't have a clear answer. I think we're going to see different buckets, right? That certain scopes are going to go into this, certain scopes are going to go into that, but know that we want to make things better and safer for patients. So we have to think about what is the highest quality, lowest risk for the patient process that we can do. And that's the way I recommend framing things. And as far as it comes to evidence, like you said, with white papers, what you want is really that firsthand evidence. So not just taking somebody's word for it, but seeing peer-reviewed literature, multiple peer-reviewed studies on a topic is always helpful in evaluating that new technology. And we do have sections within ST91 about evaluation of new technology. And I think that may be a resource for people to go to get more information along with, getting the new standard when it comes out as a whole. I think that will be helpful because there's a lot more guidance in there and new requirements related to all segments of endoscope reprocessing.
This really goes back to a theme that Justin used to almost bring up every episode, I think in season 1 and season 2 of the podcast. And it was about critical thinking. I'm asking you for the easy answer, just tell me what I need to decide. And you're telling me, well, you're going to have to think about it, Hank. You're going to have to look at the evidence. You're going to have to make a decision. That's definitely the reality in our space. And the fact that there's resources out there like what we're about to talk about with ST91 and the updates, that's encouraging because you're not out there making those decisions on an island. There are other folks out there thinking the same things, trying to work on those, at least the framework, to make those decisions in a correct way. So before we get to the 91 updates, though, we talked about detergent at the point of use. We talked about detergent in the manual cleaning process, but I want to spend at least a couple of minutes here talking about the next stage, like what happens after that manual cleaning stage to these scopes. And then we can jump into 91.
Sure. So after the manual cleaning with detergent, it either goes on to high-level disinfection, which uses a chemical, high-level disinfectant, or it goes on to sterilization, which uses a sterilant, which is a chemical. So either way, it's subjected to further chemicals in order to have that terminal step of the process completed. So disinfectants, what we use with scope has to be compatible with it. They always have to be high-level disinfectants. And you'll see within the endoscope IFUs and the literature or the validation testing from different chemical manufacturers, they'll tell you whether that disinfectant is compatible with that endoscope. Some can be used manually in a soaking basin. Actually, that's going to be one of the big updates in ST91 is we no longer recommend manual soaking in disinfectant at all. Of course, you would need to have that, I guess, as a backup, but on a routine basis, we don't recommend that manual disinfection be performed anymore. So something to keep in mind, but those disinfectants, we have ones that are also used in automated endoscope reprocessors, which is where we want the industry to go, is that anytime a scope is disinfected, it's done in an automated process because that can do a much better job than we can do manually. It flushes a higher volume of fluid at a higher velocity through that. Machine can't skip steps unless you make it skip steps, right? So it's much less variability doing it by an automated process than it is doing it manually. So that's a huge update coming out. And it's going to have a very wide impact, especially in your offsite locations, ambulatory centers, physicians' offices. A lot of facilities that in those areas are still manually disinfecting scopes in a basin of disinfectant with a lid. And think about that. It's not good for you to smell those fumes, those vapors. It's harmful. They don't always have tight fitting lids. So we do want to get away from that.
So you're very much in the know on not only these updates, but the conversations that go on behind the scenes for their development. And a big reason for that is that you've really committed such an enormous amount of your career to this conversation. And so, what was it before we get into some more of the ST91? I know you gave us a nice teaser of an important one there, but before we get into it, like, why did you dedicate your life and career to this? What really drew you in?
Yeah, so that's a long story, right? And I think we all have our own unique stories of how we get to where we are today, because I never even knew what an endoscope was when I started out in school. Or out of school, I should say. I started in the microbiology lab in a pharmaceutical company. I spent a long time there, 10 years before I moved into the medical device side of things. And I started at Olympus many years ago. and really got thrown into it, I would say. I didn't know anything about it. I knew my microbiology, of course. I'd even, taught that for a couple years. But it was quite a learning curve, and we'll say trial by fire, but it really opened my eyes to a whole side of things that I'd never seen before. And I knew that it was a difficult process, and these devices are so difficult to effectively clean and disinfect each and every time. And we're relying on the skill set of people remembering how to do these steps every time in an IFU that could be 100 pages long. So the challenge really, I think, really set me toward a career path there. And I am really motivated by learning. And I had to learn all of this. I mean, I didn't even know what an endoscope was, as I said, let alone how to clean it, how to process it. So I had to first teach myself, and then very quickly became an expert in the arena, and was recommended to be co-chair for the AAMI Working Group 84, which writes ST91, the National Standard in Flexible Endoscope Processing. That process itself started back in 2012, and then we formatted the group and started writing the standard somewhere around 2013. And I was the co-chair initially for that group, and we published the first document in 2015. And I learned so much, met so many great people involved in endoscope processing, and seeing the love and dedication from everyone really spurs you on at continuing work in the field. So I think that constant feedback and knowing that we were making such a difference in patient safety is really what's captured, my imagination and captured my love. I get very excited about endoscope reprocessing, which is something that not a lot of people can say, but it truly is my passion in the industry, and something that I've dedicated my career to making sure that it gets better for all of us, because at some point it's going to be one of us or one of our loved ones on the end of that scope. And we want to make sure that it is in the best physical shape possible that scope can be, and also from a cleanliness and a microorganism perspective, that we're not putting something into that person's body that wasn't meant to be there. So like I said, in 2015, we published that first national standard, and we've been updating it ever since then. And 2022 is our year. It is ready to go and will soon be announced. So I'm so excited for that.
Great. And hearing your story makes me feel so great that you're a part of what's going on with ST91 and being involved in that work group. And so why don't you just give us a couple more teasers then? You kind of laid out 2022 being a big year. And you already gave us one good nugget. What else should frontline technicians kind of be aware of in regards to what's coming down the pike?
Yeah. Well, there's a lot, and hopefully Hank and Justin, you'll invite me back to do a more in-depth segment on ST91, because there are so many changes that I want to just give a brief update on where we're at with it, and some things. I alluded to some things within this conversation, and I can't get to all of it today. But I think the first thing to think about is this is a brand new standard. It is not an update or an amendment like we had with the last version of ST79, where we had amendments to that. This is a whole replacement for the 2015. We have changed it from head to tail. Somebody asked me, give a number of the number of updates that are in it. I can't do that. I have no idea, because we've rewritten the whole thing. It's not just a basic update. It is a complete overhaul. So we have a lot more information in there on every topic you can think of related to endoscope processing. We have added so many references and citations to peer-reviewed literature, really with the thought that we're making these new recommendations or updates to requirements, changing some things from recommendations to requirements. How can you justify that as an end user in the facility, knowing that those changes are going to take more time? They're probably going to cost you more money. So what do you have to back up those changes? So we've put all of that into the new standard with those references to really help you formulate a plan and give you that evidence that you need to make those changes in your facility. Like I said, a lot of research, a lot of evidence. a lot of adverse event reports that we've added so that people can see what happens in the real world, what's reported to the FDA, how many scopes were used and they were damaged. They were visibly damaged before they were used on a patient, and then that patient was injured. All of that is there in the evidence, and we just wanted to make sure it was more accessible to our end users. Really thinking about building quality into the process, and you're going to get pushback on some of the things, and like cleaning verification and doing visual inspection every time and adding in time for drying. Like I said, it's definitely going to cost more money to process scopes, and it's definitely going to take more time when you're following this new standard. But we have to make sure we're doing everything for our patients. We still see tons of reports of infections related to these devices, so we know we have to do a better job with reprocessing. And that's what ST91 is about, building quality into that process based on literature and the evidence that's out there. And we've paid very close attention to our words within this new document. And I know that sounds trivial, but words mean things in the standards. So if we meant something as a recommendation only, then we captured that with a should. You'll see the word should there in whatever topic we're talking about. That's a strong recommendation based on science, based on evidence, and we included that within the standard. The other word that you'll see used a lot is shall. Shall is a requirement of the standard. So if you're claiming compliance with ST91, then a shall means that's something that you have to do, even if you don't want to do it. That is a requirement of the standard. So, things like identifying what your high-risk scopes are, performing cleaning verification every time on those high-risk scopes. There's a lot going on in there, and we've really tried to delineate by using the right words. And at the beginning of every AAMI standard is a glossary that tells you what those words mean. Should, shall, can, may, they all have different meanings. So a lot of exciting changes coming. And like I said, probably, people might not be happy about them, but I'm happy about them because it means we're going to do a better job.
Well, you don't just have an invite to come back and talk on this show. You also have an invite to join us on one of our family podcasts, Transmission Control. And when you just talked about adverse event reporting, I want to tell you it's a constant conversation on transmission control. And I think it's so great that those mod reports are getting pointed out as references, because they're just, as you said, so often overlooked. And yes, the information's there, but how much time do people have to compile all of that information and stay really educated and get their job done every day? And so you're doing a great service by including references, to all kinds of resources, not, literature, mod reports, etc. So I'm excited about this. I want to thank you for all the work that not only you, but everybody else in that work group put into essentially rewriting the standard, as you said. So not only a great job coming on this podcast and giving us, an overview and getting people excited for 2022, but also, just a great job in all of that hard work. Cause I know that's not easy, that choosing the right terminology, finding those references, putting that together and being really intentional. So I want to thank you for that.
Oh, thank you. I appreciate it.
That was Mary Ann Drosnock, Director of Clinical Affairs at Healthmark Industries. Another phenomenal interview with Mary Ann, and we got a little bit of a teaser on ST91, things that everybody needs to be on the lookout for. And I know we were just giving her a lot of props for pulling together this information. I almost feel like it's less a standard. It sounds more like a guide. It sounds like an industry guide to scope reprocessing. Not that doesn't mean it's not a standard. I just mean it's almost like a handbook and not just a standard.
It's a good study guide too. I mean, I know I used the 2015 version when I was studying for my scope certification, the CER through ISHM, and it really helped connect a lot of the dots that the textbook didn't and couldn't. It just wasn't the job of the textbook to really give that high level view. And then you drop down the standard document on it and you're like, oh, okay, so that's where that's where this plays in. Then to your point, there's some tools, there's appendix. I'm pumped. I was already excited about it. I actually like ST91 2015 better, and I like the ST79. So if everything holds true and this rewrite is as good as Marianne says, then I'm going to have a new book to put on my end table at night to read as I go to sleep, because it's really good stuff, and it helps you think through. I know I use that term critical thinking in the interview, but the more that you know about the why behind the practices, the more ammunition that you have to ask further questions in the process. To help your manufacturer and vendor partners, you just grow and grow and grow. And I agree, Justin, this is another terrific interview. And I think it's going to spark a lot of that questioning spirit that we want to cultivate in the show.
Yeah, and the movement towards standardization, as she said, many pages that have to be memorized to be able to process scopes. It's just a daunting task. And, as she said, that single-use conversation's an episode of its own. And there actually is an episode of its own coming out on transmission control. So I'll go ahead and plug that coming up here probably right around the same time. So if you're not already listening to Transmission Control, our sister podcast focused on infection control and prevention, then definitely check that one out, because there's a conversation very much in that vein. That's going to do it for this week's show. As a reminder, you can help support Beyond Clean by subscribing on Apple, Amazon, or Google Podcasts. You can also find us on Stitcher, iHeartRadio, Spotify, or just search for Beyond Clean Podcast on your favorite application. You can also access bonus content for certain episodes, but you got to go and download our smartphone app for iPhone and Android. And while you're there, why don't you give us a rating and a review, because your feedback is important to us here at the show. If you have any topics that you'd like us to cover on a future episode, just send an email to info@beyondclean.net. And on behalf of Hank and myself, thanks for listening to this week's episode of Beyond Clean.